So this is we're about to go into chapter, I believe it's 13 over protein synthesis. Today I am filming, doing this by myself. My wife who's usually helping me behind the camera. She's watching the grandkids upstairs. So there is a possibility you'll hear the pitter-patter of little ones running across the ceiling here. So please just ignore it, okay? Hopefully everybody's doing well. I know there was some confusion about how the next exam is going to be organized, but hopefully I've been able to address those particular questions. I am trying very hard to set up a Zoom discussion as soon as possible, so we'll see what happens on that. Okay. I do want to point out that the upcoming exam, which you should be able to see on the schedule when it's will be scheduled, the date for it. It's going to be a little bit harder, so you need to really kind of focus in on this just to make sure that you understand what's going on. Now this chapter is basically the translation of messenger RNA. If you remember, the actual replication of DNA is going in the 5' to 3' direction. The synthesis of the messenger RNA off of that particular gene that's encoded by the DNA is also synthesized 5' to 3'. Now when we take it to the level of translation, where we're translating that messenger RNA into a sequence of amino acids, that translation is going to translate that messenger RNA from the 5' carbon direction to the 3' carbon direction. Now as it's translating, you'll find out that there's directionality associated with the polypeptide that's being generated, meaning that it has a terminal end at the very front, which is called the end terminus, which is going to be towards the 5' end of the translation, and then the terminal end at the back end is going to be the C terminus, which is going to be at the 3' end of the messenger RNA. So keep that in mind as you're studying this, okay? This is a very important process, and I want you to think about a situation, let's say around the late 1800s to the early 1900s. About this time, scientists are starting to explore what is genetics, because of what Darwin introduced, the concept of evolution. How is that being controlled? What are the factors involved in that? There were other scientists who were trying to understand how certain diseases were being expressed and how do these things come about. And around 1900, there was one scientist who started noticing that there were certain types of these diseases had certain characteristics to them, especially one particular one where these individuals would walk in and they had a particular trait. Their urine was black. And so this was the beginning of these questions, which is the questions we're going to address initially here, were the beginning of our understanding of genetics, how it works, and how we go from a gene to a final product of a protein, which then brings about an expression of a phenotype. So that was at that particular period of time. Now the interesting part, we've gone a long way. We have gone a long way in association with understanding what this process is about. And in fact, one of the biggest questions right now is this. It's called the protein folding problem. And the question now is, how do we go from a strand of amino acids? Remember, a polypeptide, the monomer that's put together to make a polypeptide is amino acids. And how does that fold up into a very functional protein? That can bring about reactions, or it can be involved in structural elements that we need for our bodies to be able to stand here. So this is a big issue here, and because of the Human Genome Project and the things that are going on now, we're moving very fast in this direction to where we can now, pretty much, if we know the amino acid sequence of a polypeptide, we can predict and fairly accurately how it's going to fold. And this has been a big problem. the question lies is, why does it fold that way? I mean, it's interacting with, as soon as it's being generated, it's interacting with water molecules. That's what these are here. And these water molecules are pretty much interfering with its ability to fold the way it needs to fold. And so you will actually, if you watch this particular video up here with Ken Hill and his TED Talk, he will talk about how this comes about and how you got this problem with the water. But eventually what it does is it folds upright. I mean, you have these hydrophobic sequences, which these particular groups of amino acids do not like water, and they eventually fold inward so that they can get away from the water. And whereas those groups that will basically are attracted to water, they would typically fold on the outward part of a polypeptide. So there's a lot of information that we're working on now. But how did we get to this point has been the question. Well, it started, well, I'll get to that in a moment, but let's kind of make sure we understand a few comments here. So, for translation, we're talking about the translation of messenger RNA, and we're talking about codons. If you remember from your general biology, codons are actually the portions that are needed for the amino acid sequence to lead to the synthesis of a polypeptide. So we're going to talk about what the codons are. Codons consist of 3 nucleotides, and those 3 nucleotides encode for 1 amino acid. Now, there's a variety of cellular components that can be playing a role in this translation. These include proteins, RNAs, and small molecules. So what we're going to look at is the current state of knowledge regarding the molecular features of messenger RNA translation. So, the proteins that we're going to be looking at are the active participants in cell structure and function. These are the structures that are involved in bringing about the shape of the cell, the shape of an organ, the shape of the body of an organism. So they're important to that. What encodes for those proteins are the genes. So again, at this particular point, if you take in consideration, DNA is controlling everything. The sequence of the DNA controls everything about it. Me standing up here moving is controlled by the DNA. The genes encode polypeptides. This is called the protein encoding genes or structural genes. All right. These genes are going to be transcribed into messenger RNA. And the main function of the genetic material is to encode the production of cellular protein. That's the main function. In the correct cell at the proper time and in suitable amounts. So there's a lot of balancing going on here to bring about the organism. There's also a lot of balancing necessary for the organism to function correctly. So with this in mind, let's go to 1900. Particular scientist, Archibald Garrett, and he noticed that some of his patients or people that he were studying had a urine, rather than being a sort of a light yellow or yellow, it was black. And he was trying to figure out what it was. And one thing he did figure out is that this urine had a high amount of this compound called homogentistic acid, which was not found over here. So something was going on here to bring about this, because as soon as this urine was exposed to air with this homogentistic acid, it turned black. So what he did is he studied these patients that had the defects in this, and it's obvious that there's something going wrong with their metabolism. For some reason, instead of having regular urine, they're having a compound that is ending up in the urine that shouldn't be there. And so he was the first one to propose that there was a relationship between genes that they were just starting to understand are determined must be the genetic material and protein production. So this is where they're just starting to understand that genes were expressing themselves through the production of proteins. He was very interested in this alkaptone urea, which is the production of this black urine, meaning the presence of what we call alkapton, which is homogentistic acid. And so he basically wanted to understand it. Now, not only was the urine black, but their cartilage and their skin had a bluish black color to it. So he proposed, this is the way it always works in science, is that we have a situation where we make a proposal as to what's going on here. He proposed that the alkaptonuria was due to a missing enzyme. So about this time, people were exploring enzymes. They were starting to discover enzymes that they found in natural products that would carry out reactions. Now, he named the enzyme homogenistic acid oxidase. Now, he also noted, based upon Mendelian genetics, that this particular, there was an inherited pattern to this that was recessive pattern of inheritance. So he followed that. So he proposed that there was a relationship exists between the inheritance of the trait and the inheritance of a defective enzyme. Now, we understand that now, but keep in mind, they're just now starting to understand what the genetic material, and there's a lot of arguments going on at this period as to whether it's protein, whether it's DNA. So there's a lot of arguments going on. So he basically described the disease as what we refer to as an inborn error of metabolism. He was the first person to use that. And I'm sure you've heard this before, inborn errors of metabolism. I used to have a dog who had an inborn error of metabolism, which was he had a tendency to, that particular breed had a tendency to get diabetes. He was a schnauzer, a miniature schnauzer. And so as a result of that, it was expected, and so I had to give him insulin shots to help control it. So yes, inborn errors of metabolism are still seen and defined to this day. So his work was provided the first evidence of a specific relationship between genes and enzymes. That basically the mysterious diseases could be explained as inborn errors of metabolism. So that there were other ones that they were studying as well, and they said, okay, well, this is the possibility of what's causing this problem. It turns out that in urine from normal people, they did not excrete this homogenistic acid, as I mentioned before. In fact, it was metabolized. It was broken down into basic carbon dioxide and water and urine. So they reasoned that the position of a block had occurred here in that particular metabolic pathway. Something had stopped the ability to break down the homogenistic acid. And it must be, in their particular case, what they reasoned, some type of enzyme was dysfunctional. That's the oxidase. That one was dysfunctional. And as a result, instead of the homogenistic acid being broken down and broken down into smaller components, it built up. And as a result, it ended up in the bloodstream, it ended up in the urine, thus you had the black urine. So it couldn't get past that enzyme block. That enzyme was mutated and could not work on it. And we call that a block in the metabolic pathway. And then you have a buildup of the stuff like the homogenistic acid up here in this range. So basically an accumulation of chemical compounds preceding the block, preceding everything above that. So in this case, without alkaptonuria, the accumulated chemical compound is homogenistic acid. So in this particular case here, with this one, this is the homogenistic acid, and this is what it should have been broken down to, but this enzyme was dysfunctional. So there was a block here, it couldn't go through here, thus you had a buildup of this. They saw this in other particular types of things like phenylketonuria. Most children born today, they're tested for this because they don't have the ability to break down excess amounts of phenylalanine, and it causes a lot of problems for those particular children. In fact, if you look at your Diet Coke drinks and look on the side of it, it'll tell you that this has phenylalanine, and phenylketonuria should not be drinking this because it gives them too much. And there's other ones like tyrosinase. There's a variety of different things here that come into play. So there's a lot of human diseases that are caused by mutations resulting in a missing defective enzyme. So in this particular case here, if you have phenylketonuria, this enzyme's missing, you're not making any of this, and you're building up too much of that, which is going to be converted into something that could be disastrous for the individual. Thus, they try to regulate how much phenylalanine they get in that situation. Okay, so now let's jump ahead 40 years. But let's take in consideration what's going on in these particular 40 years. during this period of time, that we're looking at the biochemical pathway. So they're working out the Krebs cycle, they're working out glycolysis, they're identifying all the amino acids biochemical pathways and the productions. They're working it all through that. The geneticists are working on what are the genes, what is the DNA made out of, how is this related to producing a particular phenotype in organisms. So you have the biochemist doing that, you have the geneticist doing that, you have the embryologist trying to figure out how organisms are created, and then you have the geneticist and the biochemist and the actual embryologist arguing out what is important to the production of an individual. So all of that's going on, and at the same time, a variety of organisms are being looked at to try to figure out all these different questions that they have. And one particular group of scientists, George Beadle and Edward Tatum, in the early 1940s, they became interested in the relationship between genes, enzymes, and traits. So they looked at all three of these things: genes, they looked at the